Well, hi everyone. Welcome to the Metabolome Informatics Workshop. The work I'm speaking about today, tomorrow, and the next day spans a lot of other people's efforts. Some of them may be people you know, such as Matt Chambers, the developer in my group, Jay Holman, another developer with my team, and I've had quite a bit of help as well in putting together the work from the Scripps-specific tools, the XCMS tools, from Gary Suziak's group as well. So I'll try to let you know when I'm sharing somebody else's work with you so that you can see what's going on. This is my email address. As some of you know, I'm about to move to a new university, and so if you want to keep in touch with me, it's probably best that you make use of an email address like this one, and that will get to me. So the first of the segments that I've put together for this workshop is really an overview of Protea Wizard. It may seem a little odd to start off a metabolome informatics workshop with a discussion of a proteomics tool, but in fact, Protea Wizard was designed to be useful in biological mass spectrometry in general, not just for use within proteomics. So we'll be talking a little bit about the origins of that project and the story of how it came to be what it is. And from there, start talking in more detail about the origins of the library, the structure of what's feasible within it. How is a tool built on Protea Wizard different than a tool included within Protea Wizard, for example? And it's of course worthwhile to talk about who's made these different contributions to Protea Wizard along the way. So we'll spend some time talking about each of them. And from there, we'll also talk about a set of tools that have been built on Protea Wizard or that are included within it that are accessible to anyone who downloads the toolkit. Protea Wizard is one of the two binaries that's included on the thumb drives that should be circulating now. So if you have a Windows computer, you can just go ahead and install either the 64-bit or the 32-bit version on your computer. We do have a Mac version, or a Linux version for that matter, of Protea Wizard. The difference is that anytime you're reading a raw file format from mass spectrometry, whether it's Agilent or Thermo or Waters for that matter, you need to have access to libraries that only run in Windows. So that makes it really hard for us to build Protea Wizard for the Mac in such a way that it can read a Thermo raw file or an Agilent directory. So in the slides that you have on your laptops, there's also an agenda that spells out what happens on which days. And I'm going to try to go over that a little bit here as well. How people do metabolome informatics differs quite a lot based on what kind of data they're working with. And as a result, it's a little ambitious to say that I have one workflow that covers what people do in metabolome informatics. I'm accustomed to teaching, of course, in protein identification. And for most of the labs out there doing protein identification with shotgun proteomics, the workflows are pretty straightforward and quite similar to each other. But what I'm going to try to do with this workshop is cover the essential pieces that you would need in order to do a discovery experiment in metabolomics, where you start with raw data, figure out which ions differ, and then based on that analysis, figuring out which ions differ, following up the interesting ones to produce identifications for them. And that, in effect, covers the 3 days of the 3 afternoons of this workshop. So we start with peak picking, which is a tool, which is what Protea Wizard is going to be accomplishing for us. So if you have a profile of a peak, figuring out where it lies on kind of what m over z value you can choose to represent it and put an intensity with it as well. Be able to pull out things like chromatograms to be able to recognize the time-dependent signal as this ion appears and goes away, to recognize associated ions that are arriving and falling together. So peak picking and feature detection are fairly big deals. Feature recognition here is this problem then of recognizing related chromatograms. So perhaps you have different isotopes of the same ion. Treating each of those as an independent signal would be really foolish. So being able to recognize these associated chromatograms is important. And some of that can be rather experiment dependent. So you may have, for example, an adduct that is found along with the non-adducted form of an ion. So being able to recognize that these are associated is important. All right. From there, we have the problems of retention time alignment. So maybe you've run many replicates of the same experiment, and now you want to be able to determine whether this feature, which was seen in replicate A1, was seen in A2 or in A3? Or was it found in the other cohort in B1 and B2 and B3? So this ability to recognize that drifts in retention time may have occurred due to the chromatography of the experiment is rather important. And this is one of the features that we'll be addressing tomorrow when we talk about XCMS. And then differentiation. Differentiation is a huge challenge that shows up in all kinds of places. And it's more than simply digging through the table and saying all of the A's are higher than all of the B's. There are a lot of pieces to it that we really need to understand pretty well. And for that, we're going to talk a little bit about statistics. That comes tomorrow afternoon in the later part of the session. So we'll talk about the stats and figure out how we can determine reliably which features are differentiating two cohorts of samples. And finally, on the third day, we'll be talking about the identification of unknowns. So when we have tandem mass spectra, what can we do to make sense of them? We'll be talking about two different database resources that are out there called Metlin and MassBank. And we'll also be looking at some algorithms designed to identify metabolite species directly from tandem mass spectra. And for that, we'll be talking about a new algorithm that my graduate student, Michael Koshin, has put together called Breezy. So we'll be working through all of those examples as we move ahead. All right. So I would start by saying that Protea Wizard had sort of an organic evolution rather than being something where a bunch of PIs got together and decided it would be a really good idea to do it just like this. It was more of a case where developers in multiple laboratories found themselves working on very related problems, and they decided to team up to create something that was bigger than what any individual lab could really take on. So my first knowledge of Protea Wizard came about the time the first manuscript was being produced. So the problem that we run into is the need for a Rosetta Stone. The Rosetta Stone is an important archaeological artifact that helped us for the first time to understand how various ancient languages could be translated into each other. And in the same way, which is why the language company's name is Rosetta. So we wanted to be able to relate across different file formats that are commonplace in proteomics, across many different manufacturers' file types, and also be able to deal with all of the different file formats that have sprung up out of the weeds in the meanwhile. So we tend to think of these 5 manufacturers at the right as being kind of the major mass spectrometry vendors, but I appreciate that could be a very different list than you would see for someone working in metabolomics instead. So we didn't list Geol, for example, here, or Shimadzu, although we do have some limited support for Shimadzu. But for us, these 5 are kind of the major instruments that we're dealing with all the time within proteomics, and being able to start from any of those file formats and translate to other file formats was really hugely important for us. So we started in the usual way with the instruments we're most familiar with Thermo Fisher, for example. And the developer with my team used some of the code that had been created at the Institute for Systems Biology and refined it quite heavily to create the Protea Wizard reader for Thermo. And along the way, lots of other vendors have decided that being included in Protea Wizard is a priority. We've actually had funding support from companies like Agilent and Bruker and Waters as well to create readers that work very, very well in their file formats. And along the way, many of these companies have created new versions of their software libraries, of their data access libraries. And as a result, we needed to update our software to be able to take into account new features, things like ion mobility, drift times. If we want to be able to read drift times out of these raw data, we have to have code that's aware that these things exist. Now, along the way, there have been several community standards that have been assembled for dealing with proteomics data, and a lot of them are quite relevant to metabolomics as well. So MZXML was a format that was created at the Institute for Systems Biology as a kind of underpinning for the transproteomic pipeline. So being able to deal with MZXML files was a big priority for us. MZML was a lot slower to evolve. Now, you might ask, why would there be 2 competing standards, both built around the XML markup, around extensible markup language? Why does the community need 2 of these? And the short version is that one of these was made because the Institute for Systems Biology needed something working today. And the community effort, the HUPO-PSI effort that led to the creation of the MZML format, took a lot longer. because anytime you have 50 mass spectrometrists all trying to agree on something, it's going to take a while before all the features that they all care about are included. All right, so MZML has subsequently more or less replaced the MZXML format, but we still see lots of tools out there that evolved to read MZXML and were never updated to MZML. So being able to deal with both is important, and so our ability to read and write both of these within the Protea Wizard framework matters. In proteomics, we're frequently dealing with identification, so exporting piles of tandem mass spectra to our hard disks and being able to read these and identify them is kind of a priority. And so we see that there are lots of formats that have evolved around that. MGF, for example, is the mascot generic file. It's a format created for people who use the mascot identification software from Matrix Science. The DTA format is one that's been around with us since like 1994, and it's a kind of an ancient, archaic way to write spectra at this point. Essentially, each tandem mass spectrum gets written as a separate text file on your hard drive. So you just litter your hard drive with 10,000 1K text files. It's not a very good way to keep your computer happy with you. And PKL was a similar format derived for support of the Spectrum Mill framework for identification. And MS2 was another format that came out of the Yates Lab. So you can see there's this big proliferation of formats that have come from these. Being able to work among all of these different formats matters. Now, Protea Wizard does not directly support things like DTA and pickle files. Instead, it will create the MZML for you, and then another tool will create the DTAs and pickles from the MZML formats. So it's a chaos, as you can see. Being able to deal equally well with all of them is important. The next thing I want to talk about for you is metadata. Metadata has kind of a bad name for a lot of people in proteomics because no one wants to enter it all. But the good news is that some metadata can be automatically generated. When we run an experiment on a thermo instrument, we get a raw file that includes more than just spectral information. It will also include information like the isolation width for producing tandem mass spectra. It will also include the ion injection times, things like that. And this is true of most of the manufacturers out there. They all have pretty rich formats that are produced by the instruments themselves. MZML has a huge variety of fields that it can represent. In fact, in the last year or two, it's even picked up the ability to record things like ion drift times from ion mobility instruments. So it's a format that continues to evolve over time and give us lots of ways to store lots of information. So we have things like scan start time. You know, if you'd like to know retention times, for example, from within these files, it's valuable to be able to know. You can look right in the file and grab that out. If you want to grab out the filter string for a thermo instrument, well, here we can see that this one is, I know this is minuscule and that's sort of the point. If this was collected on an ion trap mass spectrometer, it's centroided, it was positive ion mode, nebulizing gas was used, and so on. I mean, there's a lot of information in these files. So part of our goal within Protea Wizard is to make sure that just as much of this information as possible is ported over to the MZML. So in effect, I want you to think about Protea Wizard as creating in the mind of the computer some data model representing the raw file, but that data model is represented in an MZML format, essentially. So any field that is going to appear in the MZML file has a corresponding place in memory when you've loaded a file in Protea Wizard into RAM. Okay, now I want you to think about how much information we have here on an individual spectrum and compare that to what we have in a mascot generic file. The MGF is obviously a lot simpler, but this has all kinds of antecedents out there. If you look at a DTA file, you're looking at something very, very similar. Lots of M over Z values followed by intensities. Those little pairs show up in all kinds of different formats. We have something to demarcate the beginning of a spectrum, begin ions, to specify something about the mass of the precursor. And I just want to point out that although MGF is a format that derived from proteomics, it's not immune from showing up from time to time in metabolomics. And I know it must seem offensive to some to write pep mass when it's not a peptide. But that's just how things are. So these are the examples of how rich a file format can be, or how sparse it can be, depending on what that format is intended to support. People have reworked the MGF format to play all kinds of roles. For For example, this is used in a quality control pipeline from NIST that you can get called MSQC. And in MSQC, you can create MGF files that have things like the full width at half maximum of the chromatogram from which this tandem mass spectrum was derived. But these are things that are kind of one-off formats. You cannot depend upon an MGF file that you find in the wild containing all of that information. All right, so here's our ProteaWizard Rosetta Stone. And I want to talk a little bit about what role ProteaWizard is intended to play with software that we make use of. So in this diagram, I've shown as an input either a raw file created by one of the mass spec vendors or one of these conglomerate file formats that we see, so MZXML, MZML, or MGF. Now, if ProteaWizard is reading an MGF, it has this very rich data model, but it can't populate it because those fields aren't present in most MGFs. So that is a weakness. ProteaWizard itself was written by programmers for programmers. The goal of ProteaWizard was not to produce the world's best file translation tool. It serves that role, but that's not what it was designed for. Instead, ProteaWizard is supposed to sit as the first layer, as this intermediary layer between the software that you're writing and the files. And the goal is that if you write to ProteaWizard, if you're building your tool on top of ProteaWizard, instead of having to create your own file reader, you just use ProteaWizard's because it's going to read the file for you, and there's your data model. Everything you wanted from MZML is right there in memory for you. So that's one of the initial goals. But I would take the moment to talk about the module that we're discussing here, which is called MS-Data. So MS-Data is this module within ProteaWizard that gives you the ability to envision what this file looks like in RAM without having to know where it came from. So whether you read a Waters file or an MZML from the hard drive, the data model is the same. So your software doesn't need to know what format it came from, essentially. Now, there are lots of different layers to these toolkits. So when we talk about ProteaWizard, we tend to talk about MS Convert a lot, and that's what the second lecture is all about. But within ProteaWizard, we have kind of a different stratifications of things. When you download ProteaWizard with the binaries that are traveling around on the sticks, does anybody still need the data stick, or are they? Has everyone got copies already? Oh, we're doing well. Okay, there's one copy. Someone's one in the back. Great. Okay, so we have the libraries within ProteaWizard, and these libraries are accessible to software. We have the toolkit, and these toolkits represent binary applications that are distributed with that are part of the main ProteaWizard distribution. So MS Convert is definitely the one that most people have heard of because MS Convert is the tool that we use to convert from one file format to another. We also have things like Chainsaw, which are a little less well known, but if you're doing certain curious projects in proteomics, having Chainsaw is very, very valuable for you. Chainsaw is designed to accept a protein sequence database and chop it up according to your favorite protease. So if you want to know which peptides would be produced from this database with cyanogen bromide, Chainsaw is going to help you out. All right, we have things like MS Access, which are very good for getting access to particular bits of information within the MZML file. For example, you may want to pull out the intensity associated with this particular window of retention time between this many minutes and that many minutes, between this and that M/Z value. Just pull out an intensity. MS Access will do that for you. So these are basic tools that are distributed with ProteaWizard. And then we have the projects themselves. Some projects are part of the ProteaWizard package, but are actually they're made available separately. So Skyline and Topograph are two good examples of that. Skyline is a tool that lots of people out there have been using, particularly for targeted proteomic experiments, where we want to quantify how much of these 5 peptides for this protein are found in this cohort versus that cohort. So if you're doing selected reaction monitoring projects or parallel reaction monitoring projects, Skyline is very, very handy for you. And I would also point out that Skyline is probably the project that has drawn the most attention from vendors to cause them to want their instruments supported within Protea Wizard. Because if Protea Wizard can read it, Skyline can read it. That's because Skyline is built on top of Protea Wizard. So when you're dealing with Skyline, you don't have to convert your files to MZML to begin with, because you're already running Protea Wizard as part of Skyline. Topograph was created by a great group that we work with out at Stanford. They've worked on things like protein turnover, for example, so being able to quantify these ions over time over different files is important. So these are some of the examples of things like that. I would also note that the tools within my laboratory are currently housed on our servers, but as part of this move, they will all be made part of Protea Wizard. So things like the mirror match search engine, the epitome spectral library search, and so on. These will all be made part of Protea Wizard. So they'll be distributed just the same as tools like Skyline and Topograph will be. Okay, so that's something of the structure of this. Just to give an example, there are some functions within the library that you don't get access to because, unless you're writing code that calls Protea Wizard. So if you have a peptide ion and you want to predict which fragment ions would occur if you were breaking this peptide under ETD conditions, there's no binary front end for that. But those library functions are still within Protea Wizard. So when mirror match figures out which fragments to predict for a peptide ion, it's doing so by calls to those functions within Protea Wizard. The goal here is that we want to reduce the amount of rewriting that we do within the proteomics field. That every person who writes a protein digester does so in a very slightly different way, and they may include some features well and some features poorly. We would really rather that we have a high-quality protein digester that everybody calls, because then we have known performance for how that code should behave. So avoiding the rewrite phenomenon is one of the key drivers behind Protea Wizard. All right, now I already mentioned some of the folks over at Stanford. Here we have Parag Malik and Darren Kessner. Remember that I mentioned that the developers working in these groups kind of got together and started this initiative. So I really want to throw a lot of credit to Darren Kessner and Matt Chambers. They were the lead developers that really kicked off all of this. And Kessner was the one who led the first paper on Protea Wizard in 2007, I believe. I think that's one of the papers that should show up on that thumb drive. So that effort has taken off from very small beginnings to become something that's really huge. There are lots of people out in the world who know Matt Chambers because they know Protea Wizard, because they use it all the time to convert data within their labs. But I cannot diminish in any way the impact that the collaboration with Mike McCoss and Brendan McLean have brought to this. They have been far better at recruiting vendors who want to get their instruments supported within Protea Wizard, and they brought actual money into the project to pay salaries of people as well. So this has been a really hugely valuable collaboration among these three laboratories. As for my part is mostly to tell people why they should be using Protea Wizard rather than writing it myself. But obviously I make use of Protea Wizard every day, because every time I'm using software from my lab, Protea Wizard is already embedded in it. Okay, so this is the set of folks, and I would note that all of these can be traced back to the University of Washington in one way or the other, because the Department of Molecular Biotechnology, from which I got my PhD, so I was a grad student in MBT, was a department where Rudy Aebersold worked, and Rudy Aebersold mentored Parag Malik when he was a postdoc. And Mike McCoss was a grad student, was a postdoc in the Yates lab, which was in the Department of Molecular Biotechnology as well. So you can think of all of this as stemming out of the great concentration of proteomics researchers in the Seattle area in the late 1990s. All right. Now I've mentioned MS-Convert a few times, but it's worth taking a little time to talk about it here. And as I mentioned, later this afternoon we'll be spending a lot more time with it. So essentially we want the ability to read one file format and write another. That's a pretty simple kind of capacity. We have a command line that we can use this, by which we can access this software. And we'll probably spend a little time working on the command line as we get to the later parts of this afternoon. So if we need extra caffeine, we'll find a way to make that happen. We also have a graphical user interface. So this is one of these things where I mentioned Jay Holman is somebody that I wanted to make reference to. Jay is the fellow who wrote the graphical user interface for MS Convert. So having the ability to queue up a block of files that all need to be converted in the same way, to apply filters to them and so on. These are the roles that we have MS Convert. A lot of people simply assume MS Convert is Protea Wizard. They'll simply say, I converted these with Protea Wizard version whatever. And it's okay, people understand what you mean, but the tool itself is MS Convert. Seams is a tool that, or CMS as we called it at first, is a tool for visualizing mass spectrometry data, tandem mass spectrometry or just LCMS data sets. So we have the ability to export spectra like this. The software also has the ability to apply peptide annotation to tandem mass spectra. So if you wanted to export an SVG, for example, that you were going to throw into Microsoft Word for visualizing a spectrum, you could do that. It has the ability to do all kinds of sorting, and it can work directly from raw data. So if you wanted to work from files that you just collected on your Waters instrument, you could do that. And it doesn't have any specific requirements about the data being centroided or not. It's just intended to be a viewer for whatever you have. MS Picture is one of the earliest pieces in Protea Wizard designed for the goal of quality control. Quality control is a topic that I care about a lot, and I think that many other labs out there are beginning to sense that they need to do something about quality control. If you've run experiments that spanned 10 months on the same instrument, for example, you're going to have some batch effects. The instrument is going to behave a little differently as it goes through tuning or recalibration, and being able to evaluate that graphically is one of the things that was first feasible with MS Picture. So MS Picture will read the raw file in, or the .d or whatever format you've got, and allow you to visualize those data. In this case, we're looking across, on the horizontal axis, we've got M over Z values, and down vertically, we have a retention time axis. And the color here indicates the amount of intensity that was seen at a particular spot. So you can see some things like the vertical streaks that show up all the way at the right edge of the screen. So these are ions that are obviously not separating in the LC column at all. You can also see some amount of dependence here on the M over Z value that we see as in response to time. This can reflect the increasing hydrophobicity of larger ions, essentially. So MS Picture is really valuable when you want to figure out whether your data are sane or not. Simply drawing a picture of your raw file can be very informative. So this is one of the tools that the group from Stanford, from Parag Malek, has produced. More formally, our lab has been very invested in the effort to produce good quality metrics from files. So the goal here is not necessarily to produce a low level number, such as the number of scans, though things like that are included within our metrics. The goal is to be able to produce characteristics of the files that enable us to make decisions downstream. And that is a tough combo to make. We want to be able to produce metrics that are sensible to the scientists who are operating the instruments, but that are also useful in the long-term instrument monitoring. So let's walk through those. So we start by producing metrics in various categories. We have things like the extracted ion chromatograms. We want to be able to characterize those for across the experiments. We want to be able to measure retention times to evaluate how many of certain events happen in each quartile of retention time, for example. We want to be able to evaluate the mass spectrometry. Does this file have more peaks per mass spectrum or tandem mass spectrum? Does it produce a larger number of scans or a smaller number of scans? These are the kinds of things that we want to be able to produce. Next is that we need these numbers to be evaluable over a long scale. So if you're dealing with intensity, you may have very large ratios that are produced by the intensities for a given instrument. So anytime we're talking about intensity, we're almost always doing it in a log ratio scale, which means that our numbers don't suddenly go to extremely high or extremely low values. It makes them a little more robust. Likewise, rather than say per 10 minutes how many scans did we get, we tend to think in terms of quartiles to say in the first quartile of retention time, what fraction of MS events did we observe, for example. So this gives us the ability to divvy up that time and give us a little better resolution than looking at the whole file at once, in essence. So I would give examples like these 4 as some of the metrics that we've put together. For extracted ion chromatogram, the full width at half max, Q2, meaning the median. So we can sort all of the precursor ions that are seen in an experiment, and we sort them based on their peak widths. So you have some height associated with that chromatogram. You ask what's half that height, and then ask how wide is the peak at this point within that chromatogram. You can sort all of the peaks by that, and from that pull out a median. So if you suddenly see that your peak widths are larger than they were before, there may be a problem with the chromatography, for example. So this is a pretty straightforward one, I think. Now, when we get to the RT-TIC quartiles, we come up with kind of a more obscure one. So let's imagine that we have a was this the MS? Yeah, it's MS. So we have some amount of signal that's seen in each MS scan. So we can add together all the intensities for all the peaks in that MS scan and say, this is the TIC for this particular MS scan. Now, we can ask for the whole file, what is the sum of all intensities seen in all of the MS scans? And say that that sum of everything is 100%. Now we want to ask, during the first quartile of retention time, what fraction of that TIC was seen? In the second quartile of retention time, what fraction was seen, and so on. Frequently, we want to know things like how noisy are the MS scans. If we change the injection time, are we getting more peaks or fewer, for example? So we have features like this MS1 density, which asks simply how many peaks are seen in each MS scan, and then figures out what the median of all of those would be. But this last one I kind of think of as the burger counter. A lot of people care a whole lot about shots on target. So essentially, how many tandem mass spectra are we producing in an individual experiment? If it's a lot, then we have a lot better chance of identifying lots of peptides, for example. If we have relatively few, we're not doing so well. So this frequency asks for any given minute in this experiment, what was the largest frequency? What was the largest number of scans per second that we were able to achieve? So typically we see that this value is on the order of like 10 per second if you have something like a Q-exactive firing away time and time again. The command line for launching something like this is pretty straightforward. We launch the kilometer binary. We tell it we want it to process all the raws in this directory. We specify that we want it to do the ID-free metrics because these metrics don't require that we know identifications in advance. And then we just tell it what to dump that table off to. So we've used this to process quite a few data sets, including the one that we just did for proteogenomics of colon cancer. And it's quite valuable to work through these experiments and figure out what kind of batch effects and so on we're encountering. So when we've processed large-scale experiments this way, we've been able to evaluate variability a lot more sensitively. So the experiment we're looking at right now is study 1 from the CPTAC. This was produced back in late 2006 and early 2007. And each one of these little symbol types represents a different instrument that was included in that study. Essentially, when we started CPTAC, the National Cancer Institute said, if you said you had an instrument available for our experiments, you must include it in this experiment. So there was a lot of community participation. That if we had claimed that that the LC MALDI system would participate, well, the LC MALDI showed up in this experiment as well. So here we see instruments like LTQ-B at site 65. I don't mind telling you guys, that's us, right? So So here we have each of these different experiments. So we're looking at the quality control metrics represented in principal component space. So each raw file is represented by this vector of 40 numbers produced by Quameter. Some of those metrics are correlated with each other, and as a result, they can be combined in a single principal component. So principal component analysis is dealing with all the correlations, the pairwise correlations within these metrics to figure out which ones actually explain some variance. So in this case, we're looking at the first principal component, i.e. the one that explains the most variance across the data set, and the second principal component, which explains the second most variability across the experiment. It's tempting, when you look at a graph like this, to say that being far apart is just inherently bad. For example, blue window panes here is something that we might worry about a good bit, because the blue window panes are all over the place. But I would point out that principal component analysis, if given 40 metrics, will give you 40 components back. And it may be that even though blue window panes looks really bad in terms of variability here, if we look at it in 40-dimensional space, they might have a very good grouping. So what this means essentially is that their variability is not the variability that we see across the entire experiment. That's what we can conclude. All right, so with something like this, we can visualize these quality metrics across lots of files, lots of experiments. But we can also do nice things like find outliers this way. And what I'm talking about here, yes, this was used in proteomics, but this is just as applicable to LCM SMS volumes from metabolomics. So I'm going to spend a little time here. We have these files that have been translated now into principal component space, and we want to find outliers. So one of the things that we can do is find all the pairwise comparisons that we can do. Imagine that we have 10 files. How many pairwise comparisons can we do? Does anyone know this one? Have you heard of the cocktail party problem? You have 10 people at a cocktail party. How many distinct handshakes you can get? Well, the answer to that has a little bit of sociology attached to it. So if I shake James's hand and James shakes my hand, is my shaking his hand different than his shaking my hand? It's not. Okay, so we got to divide by 2. We know that, because that's going to just cut in half all the things that we need to do. Can I shake my own hand? I could, but it would look weird. So we're going to keep in mind that if there are 10 people at a party, each person can shake only 9 other people's hands. And we've got to deal with the fact that if A shakes B, B shakes A. So that means that we're going to have n times n minus 1 over 2. All right? Now you can just pull that out of your pocket at the next cocktail party and prove you're a grad student. It's going to be great. So if you have 10 experiments from an instrument, you can say, I am going to make 45 comparisons, 10 times 9 divided by 2. So now we have these distances among all the experiments produced for this experiment, instrument, sorry. And we can ask, are the distances that we see between the vector for this experiment and the vector for this experiment, are those all producing comparable distances, or are a couple of those distances much longer than the others? So what we see is that in an instrument like Q star X at 54 here, the whole distribution is shifted off to the right, meaning that the distances among these files or between pairs of files are much greater on average. But look at this instrument down here, LTQ at site 73. This LTQ is a curious one, because we've got a whole bunch of distances right up here that are way up high, but most of them are quite low. What we found was that if you look at these distances off to the right, we see that those are all associated with one particular file that always produced high distances compared to any other file. So, in general, the experiments produced by LTQ at 73 had great comparability, but they had this one that was just way out on a tangent. So that's how you can spot an outlier, by doing these distances within principal component space from QC metrics. And these are not dependent upon which identifications you produce from these files. All right. Our goal here is that if Protea Wizard is going to do the work for you, you don't have to have you don't have to write yet another raw file reader or yet another MZML file reader. There really are enough of them in the world. So by building on top of Protea Wizard, that work is taken away from you. Certainly, Protea Wizard has a name that suggests it's all about proteomics, but it's just as useful for people working in metabolomics, and all the conversions we're going to be working with in this workshop come from it. So the basic tools that we have within Protea Wizard are accessible to software, but we have several that are accessible to people who are just willing to run things on the command line or with graphical user interfaces. So these are fairly simple tools in general that we see distributed within Protea Wizard itself, but a lot of them give you a lot more power to take on impressive projects at your desk. So hopefully some of those will be useful to you. As we continue on to the demo, we will be evaluating some of these tools in person. So I'm going to take a break here, and we will reconvene in just a moment.